People will often ask us, what are the most important findings from the ALLRT protocol? Actually, the ALLRT protocol was one of the first studies that showed us that in many people, while we get substantial immune recovery and an increase in a CD4 count with good antiviral therapy and full suppression of viral loads, not everybody recovers to normal values, and that it takes that there is a relationship with your CD4 cell count when you start antiretroviral therapy and how much recovery you get in your immune system and how long that might take when you have full suppression of viral loads on excellent antiretroviral therapy. So I think that set the stage for an awful lot of other studies that helped us identify what were those factors that contribute to either good CD4 cell recovery or failure to recover CD4 cells appropriately.
What we have learned since ALLRT was set up by our group and others, that inflammatory markers are not a good thing if they're high, and that antiretroviral therapy decreases them in many people, but not in everyone. So the significance of this study is that it provides information to suggest the possibility of even screening for these kinds of markers, and then being able to look at other treatments in addition to antiretroviral therapy that might suppress those markers and help improve outcome.
I think the ALLRT protocol also helped us identify the timing of opportunistic infections in treated patients. Most of us know that if you have advanced HIV disease and you're not on antiretroviral therapy, we have a pretty good idea of what your risk factors are for developing an opportunistic infection and at what CD4 threshold. But the ALLRT protocol enabled us to be able to tease out in people who've started antiretroviral therapy what are the factors that are associated with the occurrence of an opportunistic infection after you're already on antiretroviral therapy, and over what period of time, and what are the factors that contribute to people having an ongoing risk or having that risk go away.
Even when the virus is not detectable in blood, it is detectable in a very small number of cells. So a different set of investigators took samples stored from people who had volunteered to be in ALLRT from 1 year, 4 years, and 7 to 10 years, and looked at the cells for this, what's called latently infected virus. And we're also able to look in people who had suppressed viral loads in their blood, because that was part of the ALLRT. So they could specifically target individuals with certain characteristics to look at the evolution of their reservoir over time and how it decreased, and that is certainly important information. What they didn't show is that it decreased much more rapidly over the first year and then slower decreases over time, but that is going to help give us information that ultimately is going to help design and sort out who will be the best to target some of the early pilot interventions that are designed to perturb the reservoir and contribute to this goal of eventually having a cure for HIV.